Our next talk is about STAGETOOL, which will be shared by Oliver Meikar and Junel Solis from our Finnish node. So please take it away, Oliver.
Thank you. I'm very glad to be here and to tell you a little bit of our STAGETOOL. And I just get right to the business. I'm going to tell you first a little bit about male germ cell differentiation or spermatogenesis. That is my background, actually. As a biologist, I learned it at Turku University in Noora Kodaja's group, who I understand is also part of a meeting here. So I apologize, Nora, for everything what I'm going to say, because people do forget things. But based on this wonderful PhD time that I had, and later on, when I was studying IT, I used the knowledge what I learned, and I built a tool, not myself, but with a friends around. I built a tool that would classify spermatogenesis, and that's what I'm going to be talking to you in the second part. So let's say first a little bit about spermatogenesis. As you all know, of course, it takes place in the testes of man. It's a very complex and a process. I think it's one of the most complex tissues there are in human body. There are two very complicated tissues, brain and testes. Spermatogenesis is a continuous process. So every second, every healthy male who is attending this meeting produces more than 1,000 spermatozoa. If you look closer into the testis, you see that it contains of these fine tubules or tubuli, and the spermatogenesis takes place in these tubuli. And it takes, I don't know, can you see my mouse or not? Actually, I guess
yes,
I can.
We can.
All right. So it takes place in this tubuli, and the way how it is organized is that the germ cells, or let's say this progenitor cells, they divide slowly, and they give rise to differentiating cells, which then differentiate more and more and become spermatozoa. And then after that, they are released into the lumen of these tubules and washed away into the epididymis to be prepared for the release. So this is a continuous process, and it can be divided into these three phases. Mitotic phase is when they divide and like create identical copies of each other. And then there is a meiotic phase, which is the thing that only takes place in these germ cells, which means that the genetic material is reorganized, and the outcome of meiosis, as you know, is haploid cells, and then these then further differentiate. So it's a very complex process, very difficult to study and understand, because you cannot culture these cells normally, you cannot take them out without killing them, and so on. So as I said, if you look at this tubule again, you could draw these kind of three circles inside. So this the earliest, the slow dividing cells are the spermatogonia, and then they start to divide and undergo meiosis, and then haploid cells eventually are these red ones, and they develop into spermatozoa. And the same colors can be seen here. So if you look at the spermatogenesis, you could see that well, basically this image, it is organized in a way that you start from down left, and then you continue reading leftwards and upwards until you reach here. So you see how this spermatogonia slowly divide and then start to differentiate into type intermediate spermatogonia, type B spermatogonia, pre-leptoten, leptoten. This is already part of meiosis. Then all the genomic rearrangements takes here, crossing over, and so on. And then haploid cells arise, which then start to really change in their shape. The tails start to develop, acrosomal head. So eventually you end up with a spermatozoon. And yeah, and if you just take this one, let's say cross section out of here, this is how the tubule cross section could look like. So if you have a tubule stage 1, you could only see these types of cells. If you have, let's say, a stage 10, then you would see only these types of cells, and that makes the studying spermatogenesis complicated, and you need special people to do it. And you have to do it because otherwise you cannot understand what is going on in spermatogenesis, what genes are used at which stage, why is there infertility, and so on. And yeah, so this is a mouse testis cross section. I guess it's like 1 or 2 centimeters. And you see these are the cross sections of tubules. And if you zoom in, these are the tubules, yes? And they contain cells inside them. And every of these tubules, they are from this different stage, what I told you about. And if you look at them, they look a little bit different. Some have these like speckles inside. These are actually sperm heads ready to be released, and some don't have them at all. So this must be like later stage when the sperm is already released. And so it goes on in a continuous way. So if you want to analyze testis tissue, you need to understand the developmental stage of every tubule, and we divide them in 12 stages. And you need to understand all the cell types, like different composition of cell types in these tubules. Very complicated process. So how to actually, yeah, so it was kind of interesting assignment, not assignment, it was actually a hobby to start to try to build a model that could do this a classification by its own. So how to do it? You need human labelers for it, and you basically feed in these tappy-stained images where the nuclei are seen, and that is it. And to help the recognizing of the cells, then you also use some antibodies, like for example, peanut, PNA, so that stains acrosomal heads very nicely, so you can visualize specific cell types. Sul4, it specifically stains some spermatogonia. And for example, SMA, which is a smooth muscle actin, that specifically stains the borders of the tubuli. So if you use these antibodies, you can actually help to do this cell classification and tubule classification and labeling in a semi-automated way. So if we start with this mouse testis cross section, there are around 100 tubules, and I don't remember, like maybe 100,000 cells inside here. So what you first do, you apply, this is a smooth muscle actin, and you get this kind of tubule borders. With the help of some tuning and Photoshop and stuff, you end up having the contours. So these are now the tubule contours that can be used as ground truth on top of these images. And then you can label them, and then you cut your image into tiny pieces. And actually, we did cut it with a huge overlap. So we have several images for every cell actually in here. And then you like in our case, we cut each of these large cross sections 4, and then we used human labelers to actually go through and then say which developmental stage of this tubule it is. So, and then of course, we later ask the model to do the same for us, but this is a hard work what I do, but only specialists can do. So if we add another image on top of it, now we take the individual images of the tubules and then we do the same with the cells. So a lot of work, but it has to be done. So here, so different colors represent different cell types, what are labeled. And then in the end, you end up then a bunch of images, tens of thousands of them. You also augment them, you turn them around, you play with blurriness and other tools to make the model more robust. And then we used for the cell classification and prediction, we used the faster RCNN model, which is, I guess you have seen on images, like recognizing cars on the street and so on. And then we trained on the cells, and on the tubular context, we used then the mask RCNN to draw the masks also on top of them. And the and then there was logic to actually glue in these individual images into the larger images. So we ended up with this large image where all the tubules were predicted and all the cells were inside predicted, and that allowed you to do then a lot of statistics, which developmental tubules have which cell types and so on. So yeah, that's like a very short introduction what is spermatogenesis and how this stage tool was prepared. And I'm not going to bother you with results and conclusions. I just copied it from an article, but basically the results, like the way how this model works, it's incredibly well. We are surprised ourselves, and that was it. That's why it was important for us to have it made into an online tool that could be used then by the wider community also to help these studies. So I would like to thank from the bottom of my heart all these co-authors in here, and also Junel and his team, who is going to talk next about how this online tool actually came to be. So that's all from my part at the moment.
Thank you so much, Oliver, for giving us the background of this complex process. And I would say we can have Junel talk first, and then, of course, if we have any questions, we can put them in the chat and take them later. Thank you so much.
Thank you.
We move on to the next slide. So Oliver and colleagues approached us around September or something like that last year, around the end of September, with this stage tool that they had created. Multiple models that made predictions of these tubules and spermatozoids, or spermatogenesis, and with that they had already validated and they made a paper of. And they said, well, now it works in the command line. Can you make it more easily usable by everybody? So, bring it out to the whole world, for the whole world to use. So we sat down and we agreed to collaborate with them. So I am Junel Solis. I'm from the Turku Bioimaging, which is a part of the Finnish Advanced Microscopy Node of Euro-BioImaging. So just briefly here, we do a lot of coordination activity. We are an organization that is operated by Åbo Academy University and the University of Turku, so two universities here in Turku, Finland. We also operate an international master's degree program in biomedical imaging. And lastly, there in the bullet point, we provide this open access image analysis services and data management for academic and industry users. So for the FIAM node participation in this, the things that we had to do were to take the stage tool that Oliver and his group had made and try to figure out, well, now how do we package it so that it will work on a cloud computing resource? So we first had to standardize the inputs and outputs so that we know what to expect to get in and out. And then we decided we will develop an application programming interface server so that we can transfer data from users to the stage tool core on the web. And then we said, well, while we're at it, of course, we'll try to develop a web-based graphical interface. We'll acquire the cloud computing resources and then put it on the internet when we're ready. So here is a basic scheme of the architecture that we decided to do. Stage tool Docker container on the right side here is the core packaged. This is everything that Oliver and his team had made. And then we made this server application that talks to the stage tool core and the user's browser. And we created it in such a way now that since we have been working on it, we provisionally deployed it on this site. And don't open this on your computer because it will be very, very bad. But you can try to open it in your phone. And you can go to stagetool.bioimaging.dev. And we have included there are some demo images that you can click on, and it will make a prediction for you where the tubules are and where the cells are. So essentially, what the stage tool core deep learning algorithms were designed to do. And it also includes some information about tubule stages and the types of cells that are detected. The next steps for us here, while we have deployed it on this provisional server, is, well, of course, to make it work for larger screens, and the code is almost ready for this. In the coming days, we will update the server, so then people can start to use it on their desktop computers or laptops. But the important part here is now that it's free to use for everybody at the moment. You can go in, upload your DAPI stained image of a mouse testis cross section, and then that will make a prediction for you. Of course, we plan then to scale this up or down according to the resources that are needed. So we here at Turku Bioimaging are keeping track of how the servers are handling all of this. We tested it actually a couple of days ago, and with 2 or 3 users asking a prediction, it struggles a little bit. So it looks like we will have to scale up. But in this, we have a partner, which is the Center for Scientific Computing here in Finland. And we are working with them to see how we can scale this up and make the predictions a lot faster. Surprisingly, since this is just a prediction, we don't need any GPU resources. So we're just running this from a CPU cloud computing device. And we have plans, of course, to write a manuscript for this to detail the development process some more. So here's a list of all the STAGETOOL core developers. On the left, Oliver and his team and colleagues. And on the right, these are the Node, the Finnish Advanced Microscopy Node personnel involved with this deployment part. So I would like to mention here that, of course, Turku Bioimaging has these image data services. So you can reach us at the email address there. And with that, I'd like to thank you all for listening and for the time.
Thank you very much, both Oliver and Junel, for this fantastic talk and the tool that we can use on your computer, on your phones. And I was already doing that,